Okay, hello Sadhguru, hello. Okay, hello everyone. So friends, we are left with a little part of the DNA repair mechanism. So let's quickly finish all these DNA repair mechanisms which are left over in just 30 minutes. Okay, good morning. Okay. Sumaya, I think you missed morning class, no dear. Please kindly watch the recorded because the operon is really important concept. Okay. Good morning everyone, good morning. Whomever missed the morning class, please kindly watch the recorded of that morning class without fail. Okay. So I'll finish this particular concept very quickly, friends. Let's continue. Okay, DNA repair mechanism. So friends, if all of you very quickly recall, in the last class when we started the DNA repair mechanism, friends, we understand the different kinds of damages like depurination, deamination, oxidation, alkylation, dimer formation. Then friends, oh, that's great, Sumaya, that's great. And mutation caused by the base analogs. Later friends, we also studied the intercalating agents also, right? That's good. So after that friends, if everyone quickly recall, we started discussing about the types of DNA repair mechanisms. If you very quickly recall friends, we have already understood briefly the first type of a repair mechanism. We completed mismatch repair mechanism, we understand base excision repair mechanism, and friends, I think we have discussed these two type of the repair mechanisms. I hope every one of you remember. And friends, when we are it's very simple. I'll finish it in 30 minutes, don't worry. Okay, now in case of a base excision repair mechanism, I told to all of you friends to remember one of the most important type of the enzyme. That is what friends? DNA glycosylase enzyme. DNA glycosylase is the major enzyme which is actually involved in this particular base excision repair. And I told you now friends, please remembering enzyme names are very important friends. Later we also remembered another enzyme named as AP endonucleases. These are the two enzymes that we people remembered actually, right? Now we understand mismatch repair, base excision repair. Then friends, then the next very important repair that we have to study is nucleotide excision repair mechanism. See friends, more than 2 times the question was appeared in the part C, 1 time in the part B also the question was appeared. Friends, when you read the nucleotide excision repair, the first common point every student has to remember is nucleotide excision repair mechanism is the one which is used to repair bulky damages. I want you people to remember the statement. This is the exact statement which we got in the part B question also. What friends? Which of the following DNA repair mechanism is used to repair the bulky damages? What do you mean by the word bulky damages? I'm gonna tell you. When our DNA get exposure to the high intensity of sunlight, what is going to happen friends? There are a bulk amount of the nucleotides get damaged. So all that bulk amount of the nucleotides get has to be replaced with the healthier ones. I will explain twice and I will show you the very good animation. You will get a crystal clear idea. First of all, everyone understand what are the major enzymes that are involved in this particular nucleotide excision repair mechanism. I want all of you has to remember the enzymes we can call UVR A, B, C, D. Friends, the word UVR represents here ultraviolet rays derived. That's why we use a word UVR A, B, C, D. Kindly don't forget that. And one more very important point here friends, the UVRD, you will understand, D actually act as a helicase. And friends, when it comes to the UVRB and C, I want every one of you has to remember this very carefully. UVRB and C friends, you know what do we call them as? We will be calling them as the we will be calling them as the exonucleases. Now you have to get a doubt. What is the major difference between the exonuclease and endonuclease. Friends, we all people know very well whether it is the endonuclease or exonuclease, it doesn't matter friends. Endo or exonuclease basically will cut the phosphodiester bond. Now you look into this, I am writing here, but I am talking here exonucleases. You know what are the exonucleases? Listen my words carefully. Which can cut the phosphodiester bond at a time at two locations. I am repeating the statement. Exonucleases are the enzymes which can cut the phosphodiester bond at a time at two directions. I will tell you and I will show you how exactly the exonucleases are going to work. Please kindly don't confuse friends. Here in the nucleotide excision repair mechanism, there will be a no role of exo. Here the enzyme involved is the XC. Okay, now look into the diagram, every one of you, very carefully. All the students listen to me. Why I am going to tell, why I am calling this particular repair as the bulky damage repair. Look into the picture, all of you friends. You can see, right, there is a damage occurred here. Correct, na? Of course, yes. There is a damage occurred here. And friends, who is going to recognize this particular damage? I told you already, the enzymatic complex involved is UVR ABCD. So first friends, you listen to me carefully everyone. UVR A and B, they start scanning the DNA. They start scanning the DNA until they recognize the damage. First listen to me, I'll explain twice. There are two molecules of UVRA. How many? There are two molecules of UVRA, and friends, there is one molecule of UVRB. So what do we call it, this particular complex friends? We call it this particular complex as the UVRA2B complex. And friends, here the important statement I want you people to remember is that if this particular UVRA2B wants to active, it compulsory require the presence of ATP. So without ATP friends, UVRA2B is not going to scan that particular DNA molecule. Listen my statements very carefully friends. Now listen, two molecules of UVRA and one molecule of UVRB. Listen to me. It starts scanning the DNA. Friends, once they recognize the damage, immediately what is going to happen? All the students listen to me. Two UVRA units will get dissociated. What is getting dissociated friends? Two UVRA units getting dissociated, and how many units are left? Only UVRB is left. From here every student pay utmost attention. Now friends, UVRB is recruiting what friends? UVRB is recruiting the UVRC. Now all the students listen to me. UVRB, listen to me carefully. UVRB and UVRC together we will be calling them as the friends exonucleases. I already told you, no, they will cut the two phosphodiester bond at a time. All the students look into this. The blue color one is the UVRC and green color one is the UVRB. And friends, all the students look into the diagram. This is the area where the damage occur. What the UVRC is doing? Everyone listen, what the UVRC is doing. From the damaged site, all the students listen to me friends. From the damaged site, 8 nucleotides away from 5' direction. Friends, look into this. This is 5' direction, 3' direction. You count from here 1, 2, 3, 4, 5, 6, 7, 8. So 8 nucleotides from the damaged site towards the 5' direction can be cleaved by the UVRC. Now 8 nucleotide came out. And friends, what is this particular UVRB is doing? UVRB from the damaged site, 5 nucleotide, which direction? Towards the 3' direction it will cut. So friends, 8 nucleotides cut by the UVRC, 4 to 5 nucleotides cut by the UVRB. As a result, what is happening? 13 nucleotides can be released out immediately. Now who's gonna come into the picture? I told you na, the powerful enzyme UVRD. You only look into this, that UVRD will come and remove that entire 13 nucleotides. Then who is going to come into the picture? DNA polymerase 1 come into the picture, and replace that particular 13 nucleotides. Finally friends, ligase will come and seal the nick. What is the unique statement that every student has to remember here? The unique statement you people has to remember is that friends, hardly 1 to 3 nucleotides got damaged. That's all. Hardly how many nucleotides got damaged friends? Hardly 1 to 3 nucleotides got damaged. But how many nucleotides we removed? We removed nearly 12 to 13 nucleotides at a time. That's why nucleotide excision repair called as a bulky damage repair. Please kindly do not forget this particular statement at all. Once again, listen to my words very carefully. I'll re-explain quickly, then I'll show you wonderful animation. Now friends, this is the damage. So who is going to scan the DNA for recognizing the damage? UVRAB. 2 units of UVRA and 1 unit of UVRB with the help of ATP start scanning the DNA. The moment UVRA2B starts scanning the DNA, once they recognize the damage, 2 UVRA units will get dissociated. Then UVRB recruit UVRC. UVRB and C having Xe nuclease activity. As a result, what is happening? UVRC from the damaged site, 8 nucleotides away from the 5' direction that can be cleaved by the UVRC. So 8 nucleotides are removed out. And UVRB, what it is doing? From the damaged site, it is cleaving 4 to 5 nucleotides. Then friends, total 13 nucleotides can be removed out. That entire segment of 13 nucleotides can be released by the UVRD. Then DNA polymerase 1 comes into the picture. Then add this 13 nucleotides. Then friends, ultimately ligase will come and seal the nick. Okay, so let me show you the animation friends. By that you can understand it very clearly. Just give me a moment. See friends, these kind of things we should always remember by using the animations only. By that friends, we can remember for the longer time. Always try to remember in terms of animations. And whichever the animation I will show, you try to remember that animations, because they are very simple and you can remember for long time. Sorry. Okay. I want you to concentrate on this.
Many microbes in the environment commonly encounter ultraviolet light, which can cause neighboring pyrimidine bases, thymine or cytosine, to form dimers. Thymine dimers are the most common. Pyrimidine dimers can be repaired by several mechanisms, including a method called nucleotide excision repair. In this system, a 3-subunit exonuclease made up of the proteins UVRA, B, and C excises a patch of 12 to 13 nucleotides spanning the dimer. ATP hydrolysis provides energy for many steps throughout the process. The repair mechanism begins with the association of UVRA and UVRB and the hydrolysis of ATP. The UVRAB complex binds to the DNA as UVRA makes the initial recognition of the DNA damage. UVRA bends the DNA near the damage. With more ATP hydrolysis, UVRA loads UVRB onto the damaged DNA, after which UVRA is ejected from the complex. Next, UVRB binds UVRC. UVRC nicks the damaged strand at 2 sites flanking the damaged bases. Another complex, called UVRD helicase, strips the resulting short, single-stranded damaged fragment from the DNA molecule. The single-stranded gap that remains has a 3' hydroxyl group that allows DNA polymerase to synthesize new
Basically,
DNA.
DNA polymerase 1 will come.
After DNA polymerase is finished, DNA ligase seals the...
So finally, DNA ligase will come and seals that particular nick. So I want you people to majorly remember UVR B and C. How many Xe nucleases, how many nucleotides it is cleaving. friends, just now I have highlighted, even you seen in the diagram, in case of a prokaryotic organism, how many nucleotides we have removed? 18 to 13 nucleotides. But friends, I want you people to remember this. But if you look into the eukaryotic organism, we will remove 22 to 29 nucleotides at a time in case of a eukaryotic organism. In case of eukaryotic organism, friends, at a 5' direction, we will remove 22 nucleotides. In 3' direction, friends, we will remove 6 to 7 nucleotides. An average, in case of a eukaryotic organism, we will remove 22 to 29 nucleotides. And I want you people to remember, friends, in case of a eukaryotic organism, who will come and replace that gap? I want every student has to remember. In case of a prokaryotes, DNA polymerase 1 does the repair. I mean to say, DNA polymerase 1 replaced that nucleotide, finally ligase come and seals the nick. In case of a eukaryotic organism, please kindly keep it in your mind, this can be mediated by the DNA polymerase beta. This is the one will come and replace this particular gap. Very simple in case of eukaryotes. Okay, so friends, what will be the next type of repair? The next type of very important repair, we will be calling it as the direct repair mechanism. There is a very good animation and simple animation, but I will explain it extremely briefly, very brief, just pay attention. Direct repair. And first of all, you have to ask me, sir, why do you call direct repair? I have told you very clearly, you only observed. In case of nucleotide repair, base excision repair, mismatch repair, what we have done? Whatever the part that got damaged, we removed that part. Is it or not? We removed that part and we replaced it with the another part. But you know, friends, in case of a direct repair, what we are doing is, whatever the part that got damaged, friends, we never ever remove that part. We never remove that part. Instead of removing that part, we will repair that particular damaged part there itself. That is the reason, friends, we can call it as the direct repair mechanism. And friends, all the students, listen to me carefully. This direct repair mechanism, friends, we will be also calling it as the photoreactivation. What is the reason that we are calling it as the photoreactivation? The reason we called it as the photoreactivation, friends, just listen to me carefully. Whatever the damage that is caused by the UV light, that damage we are repairing, we are reactivating by using visible light. That is the reason only, friends, we have given the name photoreactivation. So friends, by using this particular photoreactivation or a direct repair mechanism, what kind of repairs we can repair more oftenly? We can repair cyclobutane pyrimidine dimers or pyrimidine dimers. Why does this particular pyrimidine dimers occur? First, let me explain you in one common slide. By that you will understand easily. Just give me a moment. Let me explain you an empty slide. Okay. Look into this, every one of you, very carefully. Why we are calling it as the direct repair and how we will do this and what kind of repair mechanisms, what kind of damages we can repair. Okay. All the students, listen to me very carefully, friends. Originally, see, there is a 2 thymines are present. We all people commonly know that every thymine has to pair with the adenine. So 2 thymine molecules will pair with the adenine. This is the normal phenomena. But now what is the condition? The condition is that 2 thymine molecule exposed to what? They are exposed to very high intensity of sunlight, very high intensity of the UV light, very specifically to the very high intensity of the UVB light. Because of that, what happened? These particular 2 thymines instead of pairing with the two adenines, the two thymines paired with each other, which we can call it as a pyrimidine dimer. If you look into the structure, suppose example friends, try to understand like this only. This is one thymine, this is another thymine, right? So friends, two thymines, they can form a bridge like this. A bridge like this. Originally thymine has to associated with the thymine, but what is happening? Sorry, originally thymine has to pair with the adenine. Instead of pairing with the adenine, two thymines are associated with each other. You know friends, they exactly, see, when the two thymines are paired with each other, they exactly look like a chemical. What is that chemical? We call it as we called it as the cyclobutane. So that is the reason we called this particular pyrimidine dimer as the cyclobutane photodimer, or we can shortly call it as a CPD dimer. Don't confuse. If they given the pyrimidine dimer or the CPD dimer, absolutely same. Now you can ask me one more doubt. Sir, only the pyrimidine dimers will form? Purine dimers won't form? Friends, you people has to remember, purine dimers also will form. There is no doubt about it. But the point is here, when compared to the purine dimers, the occurrence of the pyrimidine dimers are more. The formation of the pyrimidine dimers are more often when compared to the purine dimers. Okay. Now you can ask me, sir, what happens if we don't repair this particular damage? If we don't repair this particular pyrimidine dimers, friends, the only thing they will cause is the melanomas, which are a type of dangerous skin cancers. So that's why we have to repair this. Okay, how can we repair this? What is the major significant enzyme involved here? The major enzyme involved in this particular mechanism is the DNA photolyase enzyme. And every student has to remember, DNA photolyase is the first discovered DNA polymerase, first discovered DNA repair enzyme. So in a given 4-marker question, as a one of the statement also we can get, friends, first discovered enzyme is the DNA, first discovered DNA repair mechanism enzyme is the DNA photolyase only. Friends, how does the DNA photolyase looks like? I want you people to understand it. If you look into the structure of the DNA photolyase enzyme, see, just imagine this is the enzyme. This enzyme having two chromophores. I want you people to listen to me very carefully. It contains two chromophores. What are those? One we can call it as the FAD minus. Another one we can call it as the folate. FAD minus and folate. This is what, friends, exactly two chromophores present in this particular enzyme. I want every one of you understand one more important point. This particular photolyase enzyme absorb all the time great amount of the visible light. It will absorb a great amount of the visible light. And if you ask me very specifically, what color of the visible light it can absorb more, I can tell you very clearly, friends, blue color. Now this is the simple structure of the photolyase enzyme. Contain two chromophores named as one FAD minus, another one, friends, we can call it as the folate. Now I want all of you please kindly look into this, how exactly this particular pyrimidine dimer can get break by using this particular photolyase enzyme. Friends, all of you listen. First, photolyase enzyme will come and associated with that particular dimer. I have already told you very clearly what kind of chromophores it contain. It contain one chromophore named as FAD minus and one chromophore called as folate. Now let me explain to all of you how does this particular dimer can be break down. Look into the picture. You know what the FAD will do, friends? FAD starts absorbing the blue light continuously. I want all of you to understand. The FAD minus continuously start absorbing the blue light. Upon absorbing the blue light, friends, the FAD minus get excited. Listen to me carefully. The FAD minus get excited. Now the excited FAD minus release the excited electron. Once again listen to me. The FAD minus released the excited electron. Once the FAD minus released the excited electron, friends listen, this excited electron will reach this particular pyrimidine dimer. You know friends how it will break. I want every one of you pay attention towards this. It will break like this. See here, small pieces. It's keep on breaking. Who is breaking this? That excited electron keep on breaking this. You know friends, what do you can call it, this particular mechanism? We can call it a radical reversion mechanism. Don't confuse. We can call it radical reversion mechanism. I will re-explain once again. Just quickly recall. This is the structure of photolyase enzyme. Photolyase enzyme first will come and associated with this particular, what we can call it as a pyrimidine dimer. Now this particular photolyase having two chromophores named as FAD minus and folate. And what is the major function of FAD? Here FAD will come into the picture majorly, not the folate. FAD minus absorb the blue light, and upon absorbing the blue light continuously, FAD minus get excited. Upon excitation, it is releasing the excited electron. You know friends, excited electron having lot of power. Now the excited electron will go and reach the pyrimidine dimer. Now you know it starts breaking the bridge into small pieces. So that is only we can call the radical reversion. Friends, once this particular bridge got breakdown, what is happening now? The two thymines will get separated. Now two thymines get separated. Now thymine starts pairing with the adenine, and immediately this particular enzyme will get dissociated. Enzyme will get dissociated from the thymines, and friends, two thymines will finally go and pair with the adenine only. There is a no problem. Simple mechanism it is. Okay, now listen, you can ask me, sir, can you define now why we have given the word direct repair and why we have called it as the photoreactivation also? Now you can only understand. There is a bridge got formed. You only tell me, do we replace the thymines with the new thymines? No, we did not remove the thymines only. We did not remove the thymines. The thymines were there itself. We just break down the bridge which is formed. So we did not remove anything and we did not replace anything. That's why we can call it direct repair. Now, why do we call it as the photoreactivation? I told you, you only look into the picture. Why, first of all, why the pyrimidine dimer got formed, friends? Because of the UV light. Simple point. Pyrimidine dimer got formed because of the visible light, UV light. How did I repair this? By using the visible light, because FAD is continuously absorbing the light at the visible region. So whatever the damage caused by the UV light, we replaced by the visible light. So that's why we can call photovisible reactivation. Simple point. That's why we can call it direct repair or photoreactivation. I hope everyone understand. Friends, then when it comes to the SOS repair and recombination repair, these two we don't read in the molecular biology. We will read these two in the genetics chapter. That is the unit 8, genetics in the mutations. There I will explain these two very elaboratively. So you people, you no need to remember here. So in the molecular biology, I want you people to majorly concentrate on the first thing. Majorly you have to concentrate damages. All the type of damages you people has to read, all the type. Followed by majorly concentrate on the MMR, concentrate on the BER, concentrate on the NER, then concentrate on the direct repair. Remaining SOS repair, double strand breakage repair, recombination, we are going to read in the genetics chapter. Okay, so friends, that's all for now. And in today's morning, we already started the lac-operon. And Monday morning, once again, we will see the lac-operon. And Tuesday, I mean to say lac-operon and tryptophan-operon. So that can be completed. So maximum by Wednesday, unit 3 will be completed. Okay, then I have already told you friends, from Monday onwards, please listen my words. Monday onwards we will be having 2 classes till the entire month of the March. Okay, sorry, entire month of the April. So the timings will be same, 5:30 to 6:30. After unit 3 and unit 6 over, I will start the biggest unit, either unit 4 or unit 1. I will start the either unit 4 or unit 1 based on your suggestions. Let's raise one poll in the group. Whichever you say, whichever you require more, then I will go with that particular unit. And friends, every day in the Unacademy you have to attend the class without fail. Every single day, 8:30 to 9:30 a.m. you have to attend every single class. I will be starting the unit 7. Okay. So you decide whether you want me to teach unit 4 or unit 1 in the YouTube. In the Unacademy, you have to attend 8:30 to 9:30 a.m. unit 7. By that, 2 chapters I will finish simultaneously. Okay, don't forget that. Okay, Sumaya, both B and C both can act like a Xe nuclease, dear, because together only we can call the Xe nucleases. Okay, we don't call Xe nuclease, dear. We are using a word Xe nucleases because this and this will cut the phosphodiester bond at a time in 2 different directions. Okay, that's why we can call Xe nucleases. Okay, I hope it's clear for everyone. So from Monday onwards, you have to attend 2 classes, 5:30 to 6:30, 8:30 to 9:30 a.m. compulsory. Okay, friends, that's all for now. I hope everyone is clear with this. Okay, the Operon PPT will be uploaded after some time in the website. Please kindly download that. Okay, friends, that's all for now. I hope the discussion is useful. Thank you all. I'll see you in the Monday morning 5:30 a.m. sharp. Okay, thank you all. Thank you so much. Bye bye. Thank you all.